So good afternoon, everyone. Welcome to our Wednesday Endocrinology Grand Rounds. We are very happy here today to have Dr. Balamurugan talk to us about strategies to improve transplanted islet function for achieving long-term insulin independence. Dr. Balamurugan is one of our faculty in the Division of Endocrinology and Metabolism, and he is has a full-time appointment as Professor of Pediatrics and Surgery at Nationwide Children's Hospital at Ohio State University, where he's also Director of the Islet Cell Biology Lab. He obtained his Ph.D. From Christian Medical College in Vellore, one of the top medical institutions in India. He then subsequently did a fellowship in islet biology at Kyoto before coming to the United States, where he has worked at the topmost transplant centers, and particularly with respect to pancreas and islet transplant. He was at the Starzl Transplant Institute from 2001 to 2006, the Barbara Davis Diabetes Center from 2006 to 2007, and at the Schultz Diabetes Center in Minneapolis from 2017 to 2014. And we were fortunate to get him here to Louisville, where he was director of our islet transplant center from 2014 to 2020. His work has focused on advancing pancreatic islet cell isolation, purification, and methods to improve function in the setting of transplantation in both type 1 diabetes and also for type 3C. And in studying the biology of islets. He has over 300 publications. He has been an invited speaker both nationally and internationally, and mentored several undergraduate and medical students and fellows and postdoctoral students. He has been a co-investigator in multiple NIH and JDRF grants, and also was a recipient of a Jewish Foundation grant here when he was at the University of Louisville. So he has continued his work, and he will update us on the aspects of islet function and how to improve it in context of transplant. Dr. Bala.
Good afternoon, everyone. Thanks, Dr. Sri, for your kind introduction. And I thank this opportunity for, thanks for this opportunity for talk about strategies to improve the transplanted islet function. First of all, I don't have anything to disclose. First of all, why do we need a strategy to improve the transplanted islet function? If you see the transplanted islet cells immediately after transplant, they work very well. They reverse diabetes. The patient is generally insulin free after transplants. But over the period of time, the insulin independent rate decreases. Although they continue to produce C-peptide, it helps in managing the brittle diabetic condition. But our goal is to really achieve long-term complete insulin-free status. So we don't see this pattern very often, either in auto transplants or in allo transplants or even in xenotransplant model. And we need to really improve the transplanted islet function to achieve to this level so that the patient will be insulin free for a prolonged period of time. So before I start about my strategy, I would like to talk about, how we generally isolate human islet cells. The isolated islets of Langerhans are a great source of cells for transplantation to cure type 1 diabetes and for understanding the molecular pathways involved in glucose sensing and insulin secretion by the beta cells, which ultimately help to design the treatment strategies for type 2 diabetes. Isolating the islet cells from human pancreas or large animal pancreas is somewhat a specialized technique because the islets are well buried inside the exocrine compartment. So we isolate the islet cells. This is a typical islet morphology under dissecting microscope, stained with the zinc binding substance, didizone or diphenyl thiocarbazone. As you know, inside each islet, you have major 4 different cell types, including insulin producing cells and glucagon secreting cells. Once the islet cells are separated from the native pancreas, they are routinely used for islet cell autotransplants, allotransplants, xenotransplants, as well as for various diabetes research. In the case of autotransplant, it is primarily for patient undergoing pancreatectomy secondary to chronic pancreatitis, and allotransplantation for type 1 diabetic patients using brain-dead donors, which involves immunosuppression for the patients, and the xenotransplantation of pig islets to non-human primate with the aim of transplanting the pig islets to humans in near future. The pig islets give unlimited source of islet cells. You can encapsulate the islet cells so that we can transplant without any immunosuppression and all. And naturally, isolated islet cells from any donor, especially from type 2 diabetic donors, are great source of cells for our basic diabetes research. In general, for clinical islet cell transplant, we process the human pancreas in a dedicated GMP, good manufacturing GMP facility under strict FDA regulatory guidelines. Because in FDA's view, preparing islet cells is almost like manufacturing a drug. We don't want to introduce any contamination during the process. So we process the pancreas in a clean room facility, and this process involves with 4 to 5 members continuously work for 5 to 6 hours to collect islets, depending upon the nature of the pancreas. This is the typical pattern. We have different stations to process pancreas with my team in a clean room facility. And the basic method of islet isolation is the intraductal injection of enzyme collagenase to digest the pancreas to release islet cells. Basically, the enzyme collagenase digests this extracellular matrix and releases the cell pellet. Basically, the intact pancreas becomes cell clusters. This is undigested portion of the ductal cells or remaining portion of the pancreas. So once the cells are digested using collagenase and protease enzyme blend, we purify the islet cells using COB2991 cell processor with the basic principle of cell density differences between islet cells and the exocrine cells. As you see here, these are all isolated islet cells, and mostly these are all exocrine cells. So it's difficult to, collect these isolated islet cells from the exocrine population because more than 95% of the pancreatic cells are exocrine cells. So we use a Riccardi chamber to digest and use COB machine for purification. And these are all, typical purified islet cells. And we don't always get, these type of nice, pure, islet cells. We get various different degree of islet products. Some are embedded, some are fragmented. Sometimes we see large amount of exocrine cells, less amount of islet cells, different size of islet cells. So we get variations. So our goal is to really standardize this process to get maximum number of islets from every processed pancreas. So in our laboratory, these are kind of our primary focus. We do islet cell autotransplantation for pediatric patients. And, I have large experience in doing islet cell allotransplantation in University of Minnesota and University of Pittsburgh. And, islet cell xenotransplantation for porcine islets, as of now, it is commonly used for a monkey model, and our goal is to use for our human transplants. And we isolate islet cells from type 2 diabetic donors. So each process, for example, in every transplant setting, we have a different type of challenges. For example, in the case of islet autotransplant, our primary focus is to digest the pancreas to collect islets. We are not worried about how many islets we are getting from each pancreas because it's difficult to digest those chronic pancreatitis pancreas. In the case of islet allotransplantation, technically it is coming from a brain-dead donor or healthy donor. So the isolation procedure is somewhat straightforward, but we need to collect certain number of islet cells. For example, minimum of 5,000 islet cells per kilogram body weight of the recipient. If I don't achieve that number, basically it is called a failed isolation. I cannot transplant. So it comes with a different type of challenge for us. And Porcine islets, again, last year was an exciting year for xenotransplantation field because porcine heart was transplanted to humans and the kidneys were transplanted to humans. Basically, this was originally alpha-gal knockout. Alpha-gal is the primary component for hyperacute rejection in xenotransplantation model. Back in 2001 at the University of Pittsburgh, we did many transplantation studies from this alpha-gal knockout pigs, and now there is like 10 genetically modifications are done in these pigs, and that's the one going for a clinical application. And for type 2 diabetes islets or type 1 diabetic pancreatic islets, there are different type of challenge to collect good number of islet cells without damaging those islet cells for our basic islet biology research. For our islet biology study, our primary focus is how to collect maximum number of islet cells from these pancreas. That's our primary goal. Because once we collect high number, then it's easier to achieve long-term insulin independence in patients. And the second component of our research is how to provide vascular supply to islet cells. Because in the native pancreas, islets are highly vascularized organ. And once we separate from the native pancreas, we cut all the vascular supply. So we need to induce microcirculation to these islet cells before transplantation and after transplantation. And then our final approach is try to generate beta cells in the lab. Even after doing all these strategies, like providing maximum number of islets, providing microcirculation to islet cells, then finally, sometimes we may need to give more islet cells, more beta cells grown in our lab for satisfying the need of the metabolic demand, in the patients. So these are the common focus in our laboratory. And in order to improve the islet cell isolation, we use varieties of collagenase protease enzyme combinations. And even in the recent times, we also decellularize the human pancreas and collect the extracellular matrix and try to use different enzyme combination to understand how enzyme affects the extracellular matrix components to release islet cells. And when it comes to revascularizing islet cells, we are basically providing additional cell supply, basically endothelial cells, through microvascular fragment. So I'll discuss about those, how we collect those microvascular fragment. So we kind of co-transplant large amount of microvascular fragment with the human islet cells for improving the transplant islet cell function. And then the last portion, as you know, beta cells can arise from ductal cells because that is one of the theories. Beta cells generally, form from the ductal origin. So using this technology, we collect the ductal cells and try to reprogram those cells to become a beta cells. So now, why islet yield is important? Generally, the transplanted islet mass is the main factor for determining the likelihood of long-term insulin independence. Many studies reported the same, like, after doing several analysis, low islet yield leads to naturally short-term insulin independence, and more islet cells, it leads to long-term insulin independence, especially in auto transplant, because this is the only opportunity for the patient to receive islet cells in the case of auto transplants. In allo transplants, obviously, they can get a second infusion at later time. So maximizing the islet number is really important. When we isolate islet cells from either from brain dead donor, cadaveric donor for our allo transplants or chronic pancreatitis patients, we experience two different, distinct problem. Generally, in brain dead donor, we need to get a large number of islet cells. Sometimes we don't get large number of islet cells. And generally, we have a purification failure because it comes from a healthy pancreas. We get like a 40 to 50 cc of cell pellet. From the cell pellet, purifying those islets sometimes is really challenging. In the case of chronic pancreatitis pancreas, the fundamental problem is digesting the pancreas itself is a difficult problem. And we want to transplant, smaller tissue volume so that, it doesn't cause us any problem in portal vein, especially portal hypertension and other associated problems. So we get large, sometimes many times we get large amount of tissue volume from those chronic pancreatitis cases. As I mentioned, when we isolate islet cells from brain-dead donor pancreas intended for islet allotransplant, we have to collect more than 5,000 islet cells per kilogram body weight. For example, if the recipient is 70 kilogram body weight, we need at least 350,000 islet cells. Same time, if the donor is over 100 kilogram, then I need more than 500,000 islet cells. If I don't achieve that number, we call it's a failed isolation. Basically, we cannot transplant because that's the release criteria established by the FDA. So many reports looked into what are the different factors really affecting the islet isolation success. And even till today, only 50% of the processed islet preparation went into transplant, in the case of islet allotransplants. Various factors, including age, height, weight, body mass index, we call body surface area analysis, and cause of death, medical social history, all biochemical values, even the procurement technique, all various donor characteristic features affect the islet yield, cold ischemia time, and how donor is managed before the procurement, in the operating room. And those factors, plus our islet isolation factors, plus enzymes, all those factors really affect to get the maximum number of islets. So this is a classical example from the data from clinical islet cell transplantation consortium trial. 10 centers participated in the study. So it's an NIH-funded multi-center phase 3 clinical trial. In this particular study, over 324 human islet isolations performed in 10 different centers using the unified islet isolation protocol, unified donor acceptance criteria, and using all the modern latest enzyme, collagenase protease enzyme. Still, the success rate was like a 52%. Only 52% of the islet preparation went into transplant. Remaining islets not used for a transplant, maybe we would have utilized for our basic islet research. When it comes to chronic pancreatitis pancreas, as I said, the challenges are different. Isolating large number of islet cells from chronic pancreatitis pancreasis is highly essential because we need to give large number of cells. And the challenges are basically because of large amount of fibrosis throughout the pancreas. Many times it's a dilated duct or calcification in the duct, and lot of calcification crystals inside the pancreas. These factors, even they don't even allow us to really properly deliver the enzyme throughout the pancreas. So we face a different type of challenge for chronic pancreatitis case. And the two major factors affecting the islet yield, one is about the donor characteristics, which we cannot do anything. Whatever the donor comes, we have to accept the donor characteristics. We have the opportunity to improve the islet yield using a different enzyme formulation. For example, historically, we have used various crude enzymes. Historically, we started with Worthington Company, Sigma Company, Roche Company enzymes. Those enzymes were crude enzyme. In constant collaboration with these enzyme manufacturing companies, we purified the collagenase enzyme, and it helped us to get large amount of good quality islets as well as some consistency in our islet isolation. In the modern era, we have enzymes from Sarva. They are like a GMP grade enzyme, plus animal derived, no animal derived raw material in any of these enzymes. Even all these are all purified form of clinical grade enzymes are available. Although different companies, there are three major suppliers for supplying the enzymes. One is from Vitacyte, Roche, and Sarva. The enzyme is the same, collagenase is the same. It comes from Clostridium histolytic enzyme. However, they supply three different proteases. One is thermolysin, another one is Sarva neutral protease, and another one is BP protease. So when they are supplying three different proteases, it's very difficult to know which one to really use and which one is good for auto transplant, which one is good for our allo transplant. We extensively study their biochemical properties using HPLC analysis to see what type of enzymes we have it in those vials. For example, generally the enzyme, they have like a C1 and C2 collagenase, class 1 and class 2 collagenases in one vial. Sometimes those enzymes are degraded, and we see a lot of impurities even in that collagenase alone. And we carefully study their collagen degradation activity, and we study extensively the histological nature of the pancreas, and specifically staining them for different collagen type, fibronectin, laminin, and proteoglycans, and other extracellular matrix. So when we characterize these components, and we continue to interact with the enzyme companies to specifically manufacture different form of enzymes. For example, we have tested numerous enzyme combinations and successfully introduced this particular enzyme for our auto transplants. In order to do this, we tried different combinations, like combining the collagenase with the thermolysin instead of neutral protease, high trypsin-like activity enzyme. And finally, one combination was helpful in getting good islet yield for our auto transplants. And the same enzyme was not suitable for our allo transplant. So we used a different type of enzyme which has a higher proportion of intact C1 collagenase when compared to C2 collagenase. This particular combination was very helpful in getting high islet yield for our allo transplants. However, we need one enzyme. It's very difficult to switch between auto transplant, allo transplant, different type of enzyme. We continue to test many different enzyme combinations, and our islet yield was not very great, like, not more than 350,000. At least we want 400,000. After testing various enzyme combinations, still we couldn't achieve that number. And again, we continued our HPLC analysis, and we tested one particular combination based on our observation during islet process. For example, whenever we use neutral protease enzyme, we see a very solid intact islets when compared to using thermolysin. So based on this our observation, and we kind of designed the enzyme study in which we used a split lobe experiments. We test like one particular experimental enzyme in one lobe, and control lobe is our standard enzyme, and vice versa. For the next isolation, we switch the lobe to see how is our islet release pattern, from those studies. So, and based on our observation, we created one new enzyme mixture using a higher proportion of intact C1 collagenase from Wider-site enzyme, and mixing that enzyme with another company, neutral protease. That enzyme was consistently giving higher islet yield. And we tested in our auto transplant model compared to our historical enzyme, we continue to get high islet yield. And even for our allo transplant, we observed the same. And as I mentioned, in this particular study, when we created this new enzyme mixture, so there were like 249 isolation results were presented in the study. So it has 150 human islet auto transplantation and 42 allo transplantation, and 57 research isolations went into this particular study to create this new enzyme mixture. And not only islet yield, ultimately we have to check the transplant outcome. This is the beta score we calculated, based on like a day 75 basal C-peptide, stimulated C-peptide level, hemoglobin A1C, insulin requirements for the patients. And whenever we calculate the beta score from islet cells isolated after this new enzyme mixture, showed better performance in patients. And then we had a different type of challenge, because this enzyme comes from traditional Clostridium histolytic enzyme. Again, there was an inconsistency between lot to lot. So in order to avoid this lot to lot variation, and we want to improve the quality of islets, not only islet number, we want to improve the quality of islet cells. So we tested, or we created a recombinant enzyme using our standard biotechnology technique using E. coli, and we created this recombinant class 1 and recombinant class 2 enzyme. Again, we tested this in split lobe experiment and tested a different type of combinations to identify what is the best target for this recombinant collagenase. And in our recent study, we showed better islet morphology even at scanning electron microscopic level after isolating the islet cells with this recombinant enzyme. So this recombinant enzyme gives consistency, purity, as well as good viability for the islet cells. And we successfully utilized this enzyme even for our pediatric auto transplants. And we have a excellent outcome in terms of islet yield, in terms of good digestion, and the clinical outcome. Then whenever we do islet isolation from pediatric donors, we have a different type of challenge. Generally, in pediatric cases, the islets are embedded, we call mantle islet cells. The islets are not completely free. There is always a layer of exocrine cells attached with the islet cells. These attachment may give some kind of protection to islet cells, but the problem is we lose a lot of islet cells during purification because most of these cells, they kind of tend to sediment in our purification process. Because for pediatric patient, we have to provide a smaller volume of islet cells for transplantation purpose. But the purification is a very big challenge for us from the young donors. Generally, we get younger donors gives large tissue mass per gram pancreas. For example, per gram pancreas in a normal brain dead donor or chronic pancreatitis pancreas, it gives like a 0.2 to 0.3 grams of cells. But in pediatric donors, we get almost like a 50%, 0.5 gram of tissue from these cases. So we need to get the mantle-free islet cells from these pancreases. So now we refined our islet isolation method using a trisected pancreas model. Basically, we split the pancreas into 3 lobes and test different enzyme formulations in these lobes and alter the lobes for our next isolation using a miniature islet isolation setup. So this method gave us a lot of clarity for our best enzyme combination approach. So using this particular high protease approach, we get a lot of mantle islets from the pediatric donor. And we published this paper a few years ago because many of these works initiated in this campus. And at the time, one particular enzyme combination, basically it's a higher proteases, helps to get mantle-free islets. Using this approach, we try to go with a smaller tissue volume for our transplant. So we follow 2 different strategies whenever we process pancreas for our chronic pancreatitis pancreas or brain dead donor pancreas. One simple example is, in a brain dead donor pancreas, we use like an entire vial of collagenase. That's a common method most of the laboratories use. But in our approach, we dose the enzyme per gram pancreas. And we have a different strategies. For example, in the chronic pancreatitis pancreas, we may have to deliver the enzyme directly injecting the enzyme into the pancreas instead of through the pancreatic duct because of the calcification and the fibrous tissue. So this is the method we have established for processing for auto and allo cases. Now, whatever the number we get, we have a better way to collect islet cells, but even then, when we after transplanting the islet cells, they don't have the microcirculation. As you may know, islets are highly vascularized organ. The glomerulus type of minute capillaries are penetrated inside the islet cells. My islet isolation process basically it removes all these connections. So these are all the isolated islet cells under dissecting microscope. So there is no real vasculature at all. But if you see the endothelial cells or small capillaries inside the islet cells at the time of isolation, we can see all these things. The red one is stained for CD31. These are all microcapillaries inside the islet. But once we start culturing these islet cells, these endothelial cells undergo apoptosis, and there is a constant necrosis of these cells. That's why these intra-islet endothelial cells generally they don't survive. So our primary focus in our lab is to really preserve these intra-islet endothelial cell function, so that they can really re-establish once the islet cells are transplanted into the portal vein after transplantation. So this is the freshly isolated islet cells, and we developed different strategies to culture them in three-dimensional matrix with the endothelial cell growth supplement. So whenever we add endothelial growth supplement, we could form peri-islet vessels coming from these islet cells. Even at day one, we could see a few peri-islet vessels are coming. On day 7, we could really elaborate this with many peri-islet vessels. And this is the closer view of peri-islet vessel coming from an islet. And in normal islet culture, this is a fresh islet cells. And when we culture them in our standard culture approach, which we commonly use for our allotransplant, for example, in auto transplant, there is no culture step. We isolate and we transplant immediately after isolation. In allotransplant model, we need to culture the islet cells for 36 hours to 72 hours because we need to bring the patient and start day minus immunosuppression, day minus 1 immunosuppression, and then transplant the islet cells. So during the culture period, this is a typical islet morphology. There is no peri-islet vessel formation because the culture environment is not ideal for these cells to keep endothelial cells active. But we have developed this endothelial growth supplement medium, and we could form these peri-islet vessel. And when we stain them with the CD31 marker, you could see these are all like a nice vessels sprouting from the islet cells. But this approach, even this is, we conformed the peri-islet vessel with the scanning electron microscopy. This ECGS endothelial growth supplement, we cannot use it practically for our clinical transplant. Because the medium their functional capacity when we inject it into the in vivo environment, it is not really working very well. So we thought we have to supplement more endothelial cells. So where do we get the endothelial cells for our islet cells? The best sources, we cannot use any commercially available in HUVEC cells, for example. So we need to collect endothelial cells from a fat source. So we get human liposuctioned human fat, and from the human fat, we can collect stromal vascular fraction cells, and predominantly in the stromal vascular fraction cell, mostly endothelial cells. So we collect these cells and try to co-transplant with the islet cells. The problem is, we are not only transplanting endothelial, we are not only providing endothelial cells, we are also providing various other cell types, including some stem cells and all. But we want to provide only the endothelial cells. Then we modified our approach to use the peripancreatic fat, the fat which comes with the pancreas, because always there's a large amount of fat, in the human pancreas. So we standardized which area to collect the fat. And we did extensive characterization, and we did a cell separation method or cell isolation method for collecting the stromal vascular fraction cells from peripancreatic fat. So whenever we did this type of studies using human liposuctioned fat, the problem was whenever we have a human pancreas, we may not get the liposuctioned human fat. Whenever we have a liposuctioned human fat, but we don't get pancreas. But that really avoided this problem. So we directly take SVF cells from the peripancreatic fat. And whenever the cells are cultured, they form an extensive network around the islet cells and releases a lot of angiocrine factors for islet cells. Then this process there are some limitations with this approach because in pediatric patients, I don't expect large amount of fat, coming with the pancreas. And, it's a two different process. I have to isolate the islet cells, and then we have to collect the fat and collect the stromal vascular fraction cells from this fat. So instead, we wanted to really directly collect the endothelial cells from the pancreas itself, because pancreas is a highly vascularized organ. There is numerous minute capillaries are inside. So we wanted to collect these microvascular fragments, basically tiny capillaries from these human pancreas. We established a method to collect these microvascular fragments coming along with our islet cells at the time of isolation. So during isolation, whenever we isolate human islets, we not only collect islet cells, we also collect these microvascular fragments. Generally, these microvascular fragments are free-floating cells and washed away during our islet process. But we established the method to collect these islet cells. And when the cells are stained for CD31 marker, in the immunohistochemistry or immunofluorescence stain, and there is a real intact vessel with the smooth muscle alpha and the CD31 positive nature of these cells. And when we culture these cells with the islet cells, first of all, whenever we do our in vitro study, we could see they kind of tend to form aggregates around the islet cells and form a nice network. They release a lot of angiocrine factors, especially endothelial stimulating factor, and those factors stimulate the intra-islet endothelial cells to form a peri-islet vessel. So it's not only forming the vasculature around the islet cells, it is also stimulating the native endothelial cells and native intra-islet endothelial cells to form peri-islet vessel. And whenever we transplanted in an animal model, when we put these cells under in a diabetes-induced nude mouse, there also we observe the same. They form a nice vascular network around the islet cells. And our ultimate goal is to really test how these cells are functioning after transplantation in diabetes-induced mouse model. We use a nude mouse model for testing our islet cells. We induce diabetes with the streptozotocin, and after inducing with the streptozotocin, this is before inducing diabetes, after inducing diabetes, and after inducing diabetes, we make sure it is a permanent hyperglycemia to confirm whether all the islets are destroyed before we implant anything. Then we transplant the islet cells, either islet alone or islet plus our microvascular fragment. So in the case of islet alone, it took little longer to normalize the blood glucose level, and in a short period of time, they return to hyperglycemia. This is the common pattern. We see it after transplanting the islet cells in diabetes-induced mouse model. But whenever we transplanted the islet cells with MVF, we could see a quick reversal of diabetes and a prolonged reversal of diabetes. So with this approach, we are planning to transplant these microvascular fragments with islet cells for our pediatric patients. And we have, confirmed whether they establish, they kind of establish the host relation inside the portal vein. There is a clear interaction from the portal vein endothelial cells and microvascular fragment endothelial cells. This phenomenon we confirmed through dextran dye injection and other confocal microscopy. For example, in fact, we could see these vessels are really penetrating inside the islet cells. In our normal islet preparation, because before we developed this technology, whenever we transplant the islet cells, the endothelial population in that preparation is like very low. It's less than 0.5. 99% it's islet cells, some acinar cells, and some progenitor cells and other cells. But with this approach, we are enriching our islet population with more endothelial cells. In my last portion of the study, we tried to generate beta cells in some of our auto transplant cases, for example, we transplanted the islet cells from a necrotizing pancreatitis case. The pancreas is highly damaged in those cases. We don't get large amount of islets from those cases. However, we collect some ductal cells while doing the islet isolation and try to reprogram them, basically stimulate them to become a beta cell using multi-different approaches through various growth factors, adenoviral vector plus other molecular approaches to induce beta cells. Because this is in generally beta cells, they can form from the ductal cells, and these are all the ductal cells we collect during our human isolation. And in fact, in Louisville, we had the IRB to collect the resected partial pancreatectomy specimens in Norton Hospital. Dr. Gary Vitale, provided these samples to us, and we did extensive isolation of ductal cells, from these chronic pancreatitis cases. One of the primary reasons why we use this approach, as you may know, for developing beta cells, embryonic stem cells are commonly used, mesenchymal stem cells are used, iPS-derived cells are used, but we use ductal cells because we can collect the ductal cells while doing the human isolation. It's a direct opportunity for us, and we observed many beta cells are forming from the ductal cells. For example, many young donors, whenever we do histological analysis, we could see a lot of beta cells. The green is insulin positive, and the red is cytokeratin marker, basically ductal marker. So inside the duct, we could see a lot of beta cells forming from this ductal cell. This is very commonly we observed in many chronic pancreatitis cases because it's a natural injury to human pancreas. So any injury to the human pancreas, it stimulates nesidioblastosis and it forms these type of beta cells. So using this approach, we try to develop beta cells in our lab. I think with this, I would like to stop. So from every human isolation, we get islet cells, peripancreatic stromal vascular fraction cells, or pancreas-derived microvascular fragments, and ductal cells for our various studies. And our primary focus is inducing microcirculation, how endothelial cells interact with the beta cells for the regeneration, even for a normal function. So I thank the members of University of Louisville Endocrinology Department and my current team members and the Kentucky organ donor affiliates. They are the major supporter for providing human pancreas to our program. I think this is today's event code. Now, thank you. I'm happy to take any questions.
It's very trivia. Thank you. So in the past, I haven't heard much about the pediatric anxiety. Given that mostly in pediatrics, it's genetic or familial sort of pancreatitism, and assuming those are the reasons for the islet transplants. So are those pancreases different as opposed to the fibrotic chronic pancreas that we see in the house?
But surprisingly, that was my expectation. Okay. But surprisingly, we see severely fibrotic pancreas in pediatric cases. Even at the age of 4, the pancreas is almost like a rock. So my previous experience in University of Minnesota back in 2006, we did 75 pediatric auto transplants. At the time, my observation was the pancreas gross morphology is moderate to mild to moderate. But in my current place, I see more severely fibrotic cases. I think PRSS1 mutation and cystic fibrosis. Even as I said, sometimes we see like a necrotizing pancreatitis, even at that age, 4 years, 5 years.
how about preserving them for longer period?
Longer period, historically, we try to preserve the islet cells using cryopreservation technology. The technology was developed by Dr. Ray Rajat in Edmonton. So it was back in almost like 20 years ago. So because the islets, there is basically, we lose a lot of islet cells when we thaw the islet cells after cryopreservation. Because the islet cells are not a single cell or a larger organ, they are mini organelles. But in the last 20 years, this whole cryopreservation technology, there's a lot of good developments. And last year, 2022, from University of Minnesota, they published a paper in Nature Methodology of successful cryopreservation and retrieval of islet cells with high viability and function. So I think now the technology is again booming for cryopreserved types. It's basically using it's a technical vertification technology. And instead of using DMSO, they use a different approach.
So do you see many enzyme forms develop in the ducts?
Yeah.
How far do you still have some situation or is it just?
As of now, we see both actually insulin positive as well as insulin release. The comparison part is very difficult because whether it is comparable to our normal native islet cells, whether the insulin releasing capacity is similar or not. So till we are in the process of studying those ones.
Is it better to do an overnight transplant the next day and deliver the enzyme, or lose much by waiting?
Definitely, with the current methodology, we lose a lot of islet cells in culture condition. At the same time, we may eliminate some of the negative effects of enzymes or exocrine cells. They don't survive in islet culture. We get rid of those ones. Actually, that's a very good question. In Edmonton series, their initial several 50 transplants are done with non-cultured islets. Islet cells transplanted immediately after isolation. Then they also started practicing of culturing. They didn't see any major difference in terms of islet function or islet release, insulin release. As an islet biologist, generally, I like to culture them, but the culture conditions are not really great. For example, we culture the islet cells in a plain liquid culture media. That environment is not ideal. So we are developing like a semi-solid culture medium to provide a 3D matrix for the islet cells. Whenever islet cells are cultured in any type of 3D matrix like a collagen gel or agarose hydrogel or any matrigel, generally we preserve the islet cells. The problem is retrieving the islet cells from those gels is difficult. That's why we are developing this semi-solid medium. So you dilute the culture medium and retrieve those islet cells. I think that technology will really help to culture the islet cells for a longer period of time before we transplant.
Hey, Dr. Bala, I have some questions in the chat box.
Okay. What is the half-life of transplanted islet cells? So it's very difficult to give an example. So in our auto transplant model, we noticed 22 years of insulin independence and 26 years of graft function in one of the patients in Minnesota series. And the same in Minnesota series, one of our recipients, still today it's a 22nd year insulin free after receiving single dose of islet cells. But at the same time, we also noticed, only 21 months insulin free status. So an average, at least 2 years insulin independent rate is kind of consistent for over 70% of the patients. Have you tried any transgenic pig xenotransplantation under the kidney? Yes, we have used many different transgenic pigs. As I mentioned, we used alpha-gal knockout pigs back in 2001, 2002, then followed by three gene modification pigs, CD46 knockout pigs. So we used a lot of pig islet cells and transplanted under the kidney capsule in mouse model as well as in non-human primate model. To answer to this question is yes, we don't see any xeno rejection, hyperacute rejection immediately after transplant, but we continue to provide immunosuppression for surviving these islet cells.
So the kidney side for larger animals, the volume is too big.
Yeah, I think kidney capsule when it comes to larger animals, pigs in non-human primate model or pig-to-pig model, we transplant the islet cells in portal vein. But some studies are done under the kidney capsule. Even for example, in Sweden, in Karolinska Institute, Dr. Karl Groth in 1996, he transplanted the neonatal porcine islet cells under the kidney capsule of humans. Portal vein, as in some patients, he did it in kidney capsule, and he took biopsy at later stage and showed good insulin staining and a confocal microscopic image to see a lot of granules, insulin granules. But not many studies are done under the kidney capsule.
Like
kidney in adult cases, but generally in large. No, we do extrahepatic sites very commonly. But a kidney capsule is not the option. I think omentum is the main site, because in animal models like a rat, mouse, the kidney capsule is a very thin layer. It doesn't really compress my islet cells after transplantation. In larger animals, the capsule is so thick. When I put the islet cells, basically it really compresses the islet cells. So extrahepatic sites, many peritoneal cavity, omental site, they make some kind of pouch at the time of transplant for our, even my Qs did many transplants like that. When we try to inject into the portal vein, if the portal pressure is very high, we stop and we put them in the peritoneal cavity. And do you check for insulin levels in cultured islet cells? The answer is yes. Whenever we culture the islet cells, we take the islet cells and subject to glucose-stimulated insulin release, basically through dynamic glucose challenge. We expose them to low glucose, high glucose, again low glucose, or a different type of secretagogues to see how their insulin release. As well as we collect the culture supernatant from my islet cell culture and check for insulin, basically basal insulin release into the culture media. That is also we do. I think that's all the questions online.
So even though the pediatric, but still the results in pediatrics are better than adults.
Yes. Definitely, I think that is universally we see that pediatric donor islet cells, they function very well after transplant, even with the smaller number of islet cells. They are resistant to all oxidative stress, isolation stress, because we expose them to cold, mechanical stress, those type of things. I think they function very well.
That's because the vascularity.
I was just to say, is the viability because of the characteristics of the recipient, though? So, an older adult with hypertension other comorbidities versus a 15-year-old. I imagine those are factors that help with prolonging.
Definitely. Even the other way around, for example, pediatric donor islets from brain dead donor went into older donors. They worked very well. So starting from 6 months old human donor pancreas, we processed 1 year old donor, 5 years old donor. Those islet cells, they really worked very well in older recipients. Yeah, thank you. Thanks a lot.
It's very good. Leadership. Yeah. We